Today, we are going to talk about introduction to seed. A seed is the basic part of any plant. The ovules, after fertilization, develop into seeds. A seed is made up of a seed coat and an embryo. And the embryo is made up of a radicle, an embryonal axis, and one or two cotyledons. A seed is found inside a fruit, which converts into a new plant when we plant it. Hence, the seed is the most important part of the plant. Now, definition of a seed. A true seed is a reproductive unit that develops from ovules and contains an embryo, a sporophyte, and food reserves. The food is located either in embryo itself or in some external storage tissues. So, seed is the fertilized mature ovule. This means seed is a good characteristic that we can use to differentiate between fruit and other parts of the plant. If any part has seeds, then it is a fruit. For example, tomatoes, watermelon. If it does not have seeds, then it is another part of the plant. So these other plant parts are normally referred to as vegetables. Now keep in mind that there are some varieties of fruits like bananas that for her convenience have been bred to be seedless. So seeds come in all shapes and sizes. Seeds play an important role in any technology embedding, and we have to produce pure quality resistant healthy seeds for production of healthy food to satisfy our needs. So seed is the genetic vehicle of genetic innovation. The modern plant breeding and technology advances in seed industry has played a significant role in evolution of high yielding varieties and hybrid. Next is seed structure. A seed is a flowering plant's unit of reproduction and has all the materials needed to develop into uh, another plant. So seeds have three main parts. Just look at this diagram. An embryo, this is embryo. Seed coat, this brown in color, seed coat or testa, and endosperm or cotyledon. Seed coat or testa is the outer layer that surrounds the seed, and it protects the embryo against microbes and the environment. Embryo then, embryo is the end product of fertilization, and it is also known as the baby plant. The embryo will germinate and become a new plant. Then endosperm or cotyledon. Endosperm is the nutrient tissue that will feed the embryo during germination. So seeds of monocot plants have one cotyledon and seeds of dicot plants have two cotyledons. Next is types of seeds. A seed is primarily of two types, monocotyledonous seeds and dicotyledonous seeds. Monocotyledonous seeds, as I already told you, has only one cotyledon. And dicotyledonous seeds, as the name suggests, has two cotyledons. Seeds can also be either albuminous or non-albuminous. Non-albuminous seeds are the seeds do not contain any residual endosperm to store the food. For example, pea, groundnut. Albuminous seeds are the seeds that consist of an endosperm to function as a storage for the food. For example, wheat and maize. Next is monocotyledonous seeds. So first we'll discuss the structure of monocotyledonous seed. Monocotyledonous seed has only one cotyledon and one outer layer of the seed coat. This is structure of monocotyledonous seed. So different parts of the seed. First is seed coat. This is seed coat. In the seed of cereals such as maize, the seed coat is membranous and generally fused with the wall and called hull. Then endosperm. This is endosperm. The endosperm is bulky and stores food. Generally, monocotyledonous seeds are endospermic, but some, as in orchids, are non-endospermic. Next is Alluron layer. This is alluron layer. This is the outer covering of endosperm. Just look at this diagram. This is the outer covering of endosperm that separates the embryo by a proteinaceous layer. This is called alluron layer. Then embryo. Embryo is the small and situated in a groove at one end of the endosperm, right? So then scutellum. Scutellum is one large and shield-shaped cotyledon. This is scutellum. Then embryonal axis. This is embryonal axis. Here, plumule and radicle are the two ends of the embryonal axis. This is plumule and this is radicle. Then coleoptile and coleorhiza. Here, plumule and radicle are enclosed in sheaths. So they are coleoptile and coleorhiza. Plumule is enclosed in a sheath called coleoptile, and radicle enclosed in a sheath means sheath, that is known as coleorhiza. Next is dicotyledonous seed. A dicotyledonous seed has two cotyledons, and other parts of the seed include seed coat, hilum, and micropyle. Just look at this diagram. This is seed coat. This is the outermost covering of a seed, and seed coat has two layers: outer testa and inner tegmen. Hilum, this is hilum, and the hilum is a scar on the seed coat through which the developing seed was attached to the fruit. Then micropyle, it is a small pore and present above the hilum. Then embryo. Embryo, it consists of an embryonal axis and two cotyledons. This is embryo and these are two cotyledons. Cotyledons are often fleshy and full of reserve food material. Radicle and plumule. This is radicle and plumule. This is plumule. So these are the two ends of the embryonal axis. Then endosperm. It's a food storing tissue. And the endosperm is not present in the matured seed. For example, in some cases, this endosperm is not present in the matured seeds, for example, in beans and grams. Now, means these seeds are known as non-endospermous. Now, I will tell you about seed germination. Seed germination is a process by which the embryo contained in a seed develops into a seedling. There are three distinct phases of seed germination. First is water uptake by seed called imbibition. Here, embryo soaks up water from the environment and this causing the seed to swell, and the seed could then split. Next phase is lag phase. In this phase, the metabolism of the seed kicks in and the endosperm starts to break down, and the sugar are used by the embryo to grow. Then radicle emergence. In this phase, radicle grows out of the seed coat and starts to develop into a root. Once these phases are completed, the embryo will continue to grow and develop into a photosynthetic seedling that will be able to make its own food. Then seed viability and dormancy. Seed viability is the ability of the embryo to germinate. If the embryo is dead, non-functional or non-existent, seed will not be germinate. Seed dormancy then is when a viable seed is unable to germinate, even when given the ideal growing conditions for a plant. So there are three requirements that must be met before germination of seed can happen. First is the embryo must be alive. Second is dormancy must be overcome. Seeds can lay dormant for many years. For wild species, there is very important and a matter of survival. Dormancy is very important. Seed will remain dormant until the right conditions are in place. For example, a seed lay dormant when there is a drought and will only germinate when there is enough water in the environment. Then third condition is, or third requirement is, the right environmental conditions must be in place, like water, oxygen, temperature, and light. Some seeds will not germinate in high temperature or in the presence of light. So all seed will not germinate in the absence of water. Then next is importance of seed. Seed is, as I already told you, is a fertilized mature ovule together covered with seed coat. This is seed. Seed is a bridge between the two generations of a plant life. Seed is the medium which transferring character from one generation to the next generation. Seed is the vital and most important input for the crop production. Seed is used as food, feed, as medicinal, industry, or as ornamental uh, things, means have industries or ornamental value. Thanks for watching. If you like this video, make sure to subscribe for more. Thank you.